The things that tips us off that they may have been trying was in 2018, they asked for money from DARPA, and in that money, they wanted to insert the furin cleavage site, which makes it highly infectious in humans. And so if they had the idea of that, they're asking for money, they must have thought, wow, we can do this, and this is going to be a great experiment. Even our government finally at that point decided not to fund that. But what they're asking for, and this is why I think there was a holy cow moment when all of a sudden these scientists see the sequence of COVID-19, they're going, oh my goodness, didn't they ask us in 2018 to put that furin cleavage site in? And lo and behold, it's there. So what I'd like to ask, and I'm going to finish with this, and then we'll have another round if some people would like to ask some other questions, is Dr. Quay, could you sort of lay out in as simple a fashion as possible 2 or 3 items about the virus that makes you think it came from, and I don't think anybody knows with 100% whether this came from a lab or whether it came from animals, but there is some compelling evidence that suggests it could have come from the lab. And, you know, even if it was a 10% chance it came from a lab, it's another reason for us to be concerned about having oversight on this kind of research. But can you give me 2 or 3 things that this virus has that make you think it's lab versus some of the evidence for MERS and SARS that it came from animals?
Yeah, there are 3 regions: the receptor binding domain, the furin cleavage site, and this protein 8th from the left called ORF8. With respect to the receptor binding domain, if you look at what happened with SARS-1, we have the virus sequence when it first was in civet cats in the markets, jumped into a few humans. We have the virus sequence then. It started infecting more. And then we have the virus sequence when a human-to-human passage could occur and an epidemic occurred. And so you can see the progression of mutations as the virus adapted from being in civet cats and then being in humans. The first jump into humans, it had only 15% of the mutations it needed to support an epidemic. Okay, let's flip to SARS-CoV-2. When you look at the virus that first entered the human population, out of all of the changes in the receptor binding domain, there are 200 amino acids, 4,000 possible changes. There were only 17 mutations that could make it a better virus. Its receptor binding optimization was 99.5%. And in fact, one of the 17 ended up being the Delta variant. So that kind of optimization juxtaposed by the fact that there were no patients in Wuhan, 36,000 blood bank specimens tested for antibodies in 2019. Not a single patient was infected. Let's go back to SARS-1. 20% of all people in the markets were infected while the virus was practicing to set up an epidemic, 1% of the general population. So we would have expected 360 in the general population. In Wuhan, we had 0. Furin cleavage site has obviously never occurred in this related viruses, sarbecoviruses, that split from their cousins, the MERS viruses, around the time of William crossing the channel, 10,060. That was when the sarbecoviruses came. So there's never been a furin cleavage site, and the genetic sequence of it uses a code that's never been used, the CGG dimer as it's called, which has never been used before. Finally, ORF8, this protein that goes into the bloodstream and suppresses interferon response, so you're asymptomatic, and suppresses AHC antigen presentation, so you can't make good antibodies. This was the subject of 2 master's theses at the Wuhan Institute of Virology. I have found no Western scientists that worked on this location in the genome before 2019. And the protein is not present in MERS. It has a 5% homology in SARS-1. Between SARS-1 and SARS-2, there's a protein there, but it's only 5% homologous. But this master's thesis, the first one, optimized its function in suppressing interferon, symptoms of fever and chills, and suppressed its antigen presentation. The second one was making synthetic biology tools so you could move it around inside genomes.
So to reiterate, there's been no animals found that have COVID-19. When they did find that animals had the first SARS and MERS, they found it out within months. When they tested the animals in question, 90% of the animals had the SARS virus. So we haven't found any animals yet with COVID-19. And then most viruses that come from animals first aren't very infectious at first, and they infect a few humans. So you don't have a pandemic that does this. It smolders and then does this. During the smoldering phase, you find background antibodies that people have had it, even if they don't know they had it. So when they tested the background of people who were working with the animals that had COVID, they found 20% of them had antibodies to having had SARS.
SARS-1, yes, correct.
But then if we test the people in the marketplace, we're not finding that. If we look at the people in the Wuhan marketplace, we're not finding significant numbers that were positive and finding almost nobody positive from the previous year that had been ill.
No, it's 0 out of 36,000.
Thank you. Why don't we do a second round, and why don't we start, we'll go the same order. Senator Johnson?
Dr. Quay, how did we find out about the Nipah virus?
In December 2019, 5 patients at a Wuhan hospital had their specimens sent, a bronchial lavage, where they stick a throat and get it lost, to the one Institute of Virology for sequencing. The process is to amplify with a PCR process. You make a lot of copies of what's in the specimen, and you usually inadvertently make copies of what's going on in the laboratory. So the one Institute of Virology probably regrets, but they put a 55 million letter database of the background information up in the gene bank, which is the NIH's database there, of everything going on. We found 20 strange things in these patient specimens: honeysuckle genes, horse viruses. 19 of the things we found were in publications from the laboratory over the previous 2 years. So this clearly was a signal of what was going on in the lab around there. The one thing they didn't publish on was this cloning vectors of the Nipah virus. So it's in the patient specimens because it was in the laboratory at the time, not in the patients, but and they have never published on that.
Yield back.
I want to thank everybody for being part of this hearing. I don't see this as the end. I see this as the beginning of trying to understand what caused the pandemic and trying to come up with solutions. Each of your statements, which is longer than your testimony, will be available for anybody who's interested. I wanted to point out one thing from Dr. Ebright's testimony, just for those who say, well, lab leaks should be discounted, they don't ever happen. At one point, Dr. Ebright writes, the second, third, fourth, and fifth entries of the SARS virus, this was the first one, into human populations occurred as a laboratory accident in Singapore in 2003, a laboratory accident in Taipei in 2003, and 2 separate laboratory accidents in Beijing in 2004. So for people who say that it's a conspiracy theory that this could have come from the lab, they're discounting our history. The history has had these lab leaks, and so whether or not we'll ever know for 100% certain whether this came from the lab, we have had lab leaks, and we have to realize the potential danger of these pathogens. We didn't get a great deal of time into the answer. We got a little bit into the answer, but each of the scientists we asked today were asked to let us know how we could better supervise or oversee this kind of research. And the interesting thing to me is I think they all worked independently, but they came up with basically very similar solutions. An independent body outside of the funding organizations or those receiving the funding to make the recommendations. Something akin to an independent agency like a nuclear regulatory agency. In fact, I've already been using the analogy when people ask me and say, well, what's this like? And it's essentially, we don't let anybody sell centrifuges to Russia or centrifuges to Iran. There are rules on the export of things. And I think Dr. Esfelt in particular has talked about the security aspect of this. What I would really like to come of this, and I mean this sincerely, is I would like to have a bipartisan bill that comes forward for better oversight. Maybe it's not oversight of gain of function, but maybe includes things that some people consider to be gain of function. Maybe it's more general of pandemic viruses. There's a lot of ways we can discuss it, but the bottom line is I don't think the people doing the research are able to adequately and objectively regulate themselves. And I think having 1 million people die, there should be bipartisan curiosity in this, that we should be able to move forward. So my hope is that your suggestions that you've taken the time to put in writing, you've taken the time out of your busy careers to come here, that these suggestions will become legislation. If we can get a bipartisan bill to come forward, what I'd like is that our people who help us write the legislation can communicate with the 3 scientists here. We're willing to hear from a dozen more scientists, anybody who wants to. I want scientists to be involved in this. But I do think that ultimately the people making the judgment shouldn't be from one small field of science. So some have said, well, none of the 3 scientists there are virologists. Well, I don't have a problem with virologists being part of this, but I do have a problem with them all being virologists. The same way I have the problem with behavioral science being approved for funding by all behavioral scientists. I think that there need to be people who understand science on this. I think there also need to be people on the committee, as Dr. Esfeld has mentioned, that understand bioterrorism and biosecurity. And I think it should be a mixture. I mean, this is something we can talk to the scientific community. And I don't think an absolute ban is what we want. We want is a better oversight of this, but we can't have something where 3 projects have been looked at in the last 7 years. I mean, that means they're not looking. And the fact that they didn't look at what went on in Wuhan, and then some of the folks I asked in committee about this were saying, oh, our scientists looked at it and approved it. Even that's not really true. They didn't look at the research. They just ignored the research. It didn't go before the committee. They haven't been honest. But if we want trust in public health and trust in government and trust in science and trust in research and trust in the NIH and trust in the grants that we give to our universities, billions of dollars, we need to have transparency and honesty. We can't have a committee where the people are cloaked in secret. I mean, what is this? I mean, this is completely insane. So I think we've made some progress. I want to move forward, and I, for one, am open to work with any Democrat in the Senate to make this a bipartisan bill and to make it an even-keeled thing where all the voices are heard, that we don't rashly create any legislation that would hamper science, but that we create something that would have oversight that might save lives. I truly think that 1 million people died in our country, 6 million people died, and I think it was from a lab leak. And I think it's something that we need to have precautions against, because if this gets in, and I think it was accidental, by the way, I mean, no, but I think if we don't do anything, what if this gets in the hands of somebody who actually really wants to harm America or the world, or just some psychopath? What could happen? So right now we're doing nothing and have changed no behavior. We've had this pandemic, and we have changed not one bit of behavior. So I think it's about time we do get together and that we're all curious, and that we don't make this about Republicans and Democrats. We make this about how we as a people come together to try to make this world a better place. Thank you all for appearing.